Hello, friends. Since you're all in quarantine and I have a captive audience, I wanted to clarify some of the things that you've been asking me repeatedly about COVID. I know we see a lot of things on TV and social media, and experts are telling you things, but I wanted to share this with you for what is what. First, let me introduce myself. For those of you who don't know me, I'm a physician. I trained in public health and I work in health IT, population health and analytics. I'm going to clarify that I'm not an expert in COVID-19, and neither is anybody else at this time. Really, no one else is. But I will give you the facts as I see them, and you can make your own decisions on predictive modeling and where this might be going. One of the three words I was told by a mentor while being humbled in the field of medicine is to say, I don't know. And given that this is a new disease and we haven't really seen it before, we don't know is going to be a very common answer. But like I said, before too long, let me get to some of the questions that you've posed to me and let me try to answer them to the best of my knowledge and what's out there. I also just wanted to say that we should move past from this idea of fear to fact. It's not about what may happen anymore. It's all about what is happening. It's a reality now, and we cannot wait any longer for what if it happened to me. We are showing a very deep vulnerability, and we need to arm ourselves with the correct knowledge on how to deal with this. Having said that, I hope that this being a trial run of today's recording, I will be able to do a few more segments, either by myself or with a couple of friends who are also involved in this, and try to answer some of the basics, to the guidelines, to the treatment, to whether you should be using a mask or not, things of that nature. Today, let me just address a very few basic questions that came my way, as in, what is this? What is a coronavirus? So a virus is basically an energy-less floating life form or an organism that thrives inside another organism's cell. The nucleotides that make up this protein, they're arranged in a different fashion as humans, but they can replicate inside human cells. Coronavirus are the same viridae that cause 15% of the common cold cases every year. The rest are caused by other viruses. Coronavirus is an RNA virus, which means that its genetic material contains information that can actually make millions of clones of the original virus. Replication of the genetic material occurs when the virus takes control of a host cell's synthetic machinery, and that instructs the machinery to build enzymes and new viral structures and proteins. Then they copy that genetic information enough times so that a copy can be placed in each newly constructed cell. Finally, they leave the host cell. They invade the virus. The invading virus blocks the synthesis of any host cell DNA, any RNA or proteins, such as inside humans. This feature of the host cell to construct only viral proteins and copies of the viral genetic material is unique to viruses. So in a nutshell, whenever a virus replicates, it must complete a couple of steps. So it gets absorbed, or in viral terms, it gets adsorbed. It penetrates. Then an uncoating of the virus from its middle structure, the nucleus structure, happens, and the capsid or the coat uncoats. Then it goes inside, it starts synthesizing, and it assembles viral proteins inside the cell, and it inhibits the host cell DNA and RNA, and so on and so forth. And then once that happens, it releases these virion particles into the host cell, either by chipping off the cell's membrane or by sending off buds. So what happens as a result? What happens as a result of all of this is a couple of outcomes. There could be host cell death. So when it shuts down the host cell's functions, which is almost always commandeered by the viral replicating, virus replicating, that causes the death of the cell. And other times it transforms the cell itself. It can induce things like oncogenes, for instance, which can cause uncontrolled activity of the cell and its growth, and uninhibited cell growth, and cause things like cancer. Or it can, we've been hearing a lot of people that are not showing any symptoms, in which case you can call it an asymptomatic person or a latent infection, which actually means either it survives in a sleeping state, but it's not producing any clinically significant symptoms for you to say that you're showing any symptoms. Or it can, at other times when you're immune compromised, result in reactivation of the symptoms. An example of that is, say, for instance, a herpes virus, that, if you've ever had it, it comes up as shingles. The other thing that it can do to you is it can result in a chronic slow infection. So what are its outcomes? I've already told you now. Where are we? What are the origins, and how is it evolving? So like I said, its origins could be inside the host cell of animals, such as bats and pangolins and what have you, like we've been hearing these reports. It looks like this RNA virus took a jump from the animal friends to its human cousins. And, well, what does that mean? It means that once it's inside the human body, its RNA is using that machinery to replicate inside human cells, and now it's capable of not just animal to human transmission, but now it's capable of human to human transmission. So a couple of people have asked me, we've seen SARS, we've seen MERS, and people are saying those are also coronaviridae. And we've seen avian influenza, which is the H1N1 viruses and H5N1 viruses that cause those epidemics of flu. What makes the coronavirus so transmissible and lethal? Let me try to attempt to answer that for you. Viruses try to jump host cells all the time. The more species they can infect, the more they can replicate eternally. So most of the viruses, such as RNA viruses, and those that exhibit some form of plasticity, meaning they can infect more than one kind of animal, are the ones that are most likely to jump. In this virus, the case of the coronavirus, the prevailing theory is that the virus was mostly a bat virus, but it managed to combine with the spike proteins from some pangolin viruses. And in an environment when bats and pangolins came together and had contact with humans and with each other, the new virus then found its way into the human, and this phenomena is commonly seen, say for instance, in 20% of all kinds of zoonotic or animal transmission cases. Other things that make it optimal for spreading is it spreads heavily when hosts are asymptomatic. So for instance, SARS and MERS, they only had very high viral titers when people were already quite sick in a hospital and not walking around in crowded markets. It was very non-specific, and it had very non-frightening symptoms at first. So this is very unlike, say for instance, Ebola, where inhibition of the coagulation, which is your blood clotting mechanism, causes people to bleed profusely, from different orifices, because that's what it's causing, hemorrhaging inside your body. So if you know that something is very wrong and it sounds like hemorrhaging, it's probably Ebola, so you kind of know you want to stay away. But if it's just a little dry cough that comes on at first, which is what is seen commonly with the coronavirus, it's not too terrifying, and you probably are unlikely to keep your distance. And it has a respiratory or a droplet mode of infection, of transmission. And since we breathe air all the time and we're normally in close contact with people, it's very easier to spread this virus very widely, as opposed to something that might be bloodborne, or something which requires close contact. So we talk about the R, the R is nice in this case. The R is the number of people that it infects, the R naught, and that's kind of nice. It's like about 2.4, keeps humming along, but it doesn't burn so hot like to say, that it's kind of causing much damage. But what it does do is if one person gets infected and the R naught is 2.4 or 2.5, which means it has the capacity to infect that many people, it actually can, transmit to so many that it keeps replicating. And at the end of it, what we're seeing is sepsis in people and complete organ failure and death. So if it can make copies and it is causing virulence, it is also leading to death as an outcome of its virulence. And that's what tells us that it can be lethal. But it also tells us sometimes that, an R of 2.4, 2.5 may not be as lethal. So that's one aspect of it. Another very basic thing that I've been asked is, I read about all these strains of viruses. Does that mean that I get infected that many times if I come across it? Is it mutating? Is it doing it so fast that I'll never get to a vaccine? Well, first of all, a vaccine is preventive. And secondly, vaccines take time in development. We hear that there have been vaccines that have been tested on the animal model, but we still don't have any results on the human model. So we're waiting. And a vaccine is not curative. A vaccine is only preventive, just like at the beginning of a flu season, we'll get a vaccine and we hope that it'll give us some immunity against the more severe symptoms of flu if we were to catch it, or prevent us from catching it at all. Again, lots of different strains out there, 8 or 9 different strains we're seeing. Is it mutating very fast? No, it's not mutating very fast. I think for today, I'm going to stop the session because we're already 12 minutes into information overload, as people have been saying. If you are able to follow this along and you think you want me to continue with more information, let me know. Leave a comment and I'll be sure to get back to you soonest. Bye.